Okay, so we'll start seeing folks come streaming in at the bottom there where it says participants, Dr. Amara. Welcome, everyone. We'll get you started in just a few minutes. Okay, I'm going to get started in just about 30 seconds or so here, folks. So anybody coming in, we'll get started in just a minute with our keynote address and the sharing of our winners for the Harry and Linda Serene Leadership Essay Contest. Okay, I'm going to get started. Good morning, everyone. It's a pleasure to be here with you today. I'd like to welcome you to the 6th Annual Strike a Spark Conference for Research, Creativity, and Scholarship. My name is Dr. Azadeh Masalan-Block, and I'm the Director of the Center for Undergraduate Research here at California University of Pennsylvania. This year's theme for the conference is Staying Connected Through Leadership in Research and Creativity. The pandemic has brought much adversity to the academic world, and the ability of our student and faculties to connect has been widening with the Zoom gap, and our feeling of a campus community has certainly been strained. However, despite all of that, we have over 120 students participating in this year's conference, and our planning committee has done wonderful things to make this event come to fruition this year. We hope that this event truly strikes a spark for our campus community to return stronger than ever to what we hope will be a more typical fall semester at CalU. Today, we have 3 important things on our agenda. We have 2 outstanding speakers, both on the cutting edge of research and creativity in their respective fields, and another positive light to shine in terms of sharing the winners from this year's Serene Leadership Essay Contest. We'll begin this webinar today with an address from Dr. Susan Amara, who is the Scientific Director of the National Institute of Mental Health's Intramural Research Program at the National Institute of Health. Dr. Amara joined the NIMH after serving as the Thomas Detry Professor of Neurobiology and Distinguished Professor at the University of Pittsburgh School of Medicine. Work in her laboratory has focused on the structure, function, and cellular physiology of neurotransmitter transporters, including the biogenic amine transporters, major targets of psychostimulant drugs and antidepressants. Amara received her BS in Biological Sciences from Stanford University and a PhD in Physiology and Pharmacology from the University of California, San Diego, and previously held faculty positions at Yale University School of Medicine, at the Volum Institute in Portland, Oregon, and as a Howard Hughes Medical Institute investigator at Yale and in Oregon. Her awards include the Burroughs Wellcome Hitchings Award in Drug Discovery, the Society for Neuroscience Young Investigator Award, the Aspet John Jacob Abel Award, a McKnight Neuroscience Investigator Award, a Merit Award from NIDA, a NARSAD Distinguished Investigator Award, the Julius Axelrod Award from Aspet, and the Julius Axelrod Prize from Society for Neuroscience. Amara is a member of the National Academy of Sciences, a fellow of the American Association of the Advancement of Science, and past president of the Society for Neuroscience, and president-elect of AAAS. So Dr. Amara, thank you so much for being with us. I'd like to turn the webinar over to you to present and share with us some insights for our students and faculty today.
Thank you, Ozzy. It's great to be here. I'll share my screen and put my presentation up. Here we go. So I want to start by saying what a great series of talks I heard this morning. And I was particularly gratified because so many of them emphasize things that I actually, and you'll see throughout my talk, that I actually care a lot about as well. And I enjoyed the idea that many of you are very passionate about things like stress reduction, diversity, and I also just appreciated the service that you all do and the volunteer service that you've done in terms of all of these areas, including management of addiction. I really did enjoy all your talks. So the title of my talk is really a quote from Yogi Berra: "When you come to a fork in the road, take it." And when I was asked to give this talk, I was asked to really talk a little bit about how I got to where I am. So as you'll see, I'm going to start at the very beginning, and we'll tell you a little bit. And what I hope is that a story, we all have stories, and certainly they're all unique. And what I hope that this really tells you is there are really many, many paths to a career in science and research. And there are also many ways one can contribute to the whole enterprise of research, no matter what you do. So with that, I will start out at the very beginning, which is my grandparents. So I'm going to start with my maternal roots. This is a picture of my grandmother. Her family moved to British Honduras to farm bananas, and there she is, actually paddling a boat that they used to carry the bananas in up and down one of the rivers there. And she was married in British Honduras to she was American and he was British, but then she moved to a cane plantation in Cuba. It was right next door to Fidel Castro's family's farm. So they knew his family. And my mother, who's pictured on the extreme right over here, and maybe I should get the pointer, pen, laser color, green. Can you see that, or is the laser pointer showing up? Yeah, good. Okay, so anyway, my mother ended up going to being sent to British boarding school in Jamaica, another island. And you might ask me why all this matters. Well, it turns out my grandmother, while in British Honduras, collected bugs and snakes. And I was always fascinated, and she had jars right next to the canned apricots of these really fascinating organisms. And I have to say, when I was a child going up to visit her, it was fun because she had great stories about exotic animals, and she had this collection of bugs you can't believe. And shown here is a whole series of different organisms that my lab has actually studied over the time that I've done research. And what you'll find is that many of those organisms are remarkably similar to things that my grandmother had in her collection. So the second part of my family is paternal roots. My grandmother, shown here, Francesca, and this is my father, Dominic. They came from Sicily to Boston. And from Boston, they went back to Sicily, where my father became an apprenticed carpenter. Mussolini came in, and my family moved out back to Boston because my grandmother didn't want to be under the Mussolini regime. And so rather than being a carpenter, my dad ended up going to college, and he became an engineer and on the faculty at MIT. So there was another aspect, which, and certainly from my father, I learned the importance of science and the value of science and mathematics. Although I ended up going into biology. My parents married in New York, and I grew up as a pretty normal kid in the suburbs of Los Angeles, the San Fernando Valley. And I kind of can trace back my interest in science. I was in the first generation of women and on either side of my family to go to college. But I had a number of really early epiphanies that made me really interested in science. And it also made me aware of some of the pitfalls. So my first epiphany was that you can be a scientist and have a job. And this was because I was sent to a field trip to a drug company from my junior high school. And I saw people working, doing experiments, studying how drugs worked. And they were all employed. And in my family, it was really important that you have a job. And so I thought, wow, I can be a scientist and have a job. So that was good. And in fact, that really sparked my initial interest in pharmacology, which is in fact the area, neuropharmacology has been the area of research I've done over the course of my career. I also learned that it would be harder if you're a girl. In ninth grade, I was sent to a lecture by a representative from NASA who was talking about science careers. And when he was asked by a rather brave young woman or young girl in the audience whether girls could actually become astronauts or participate in NASA, and he said, "Oh no, you really, we don't think you can." And he went on to say something like, first of all, it would be really hard to deal with all the bodily functions that women have. And would you want to be on a mission that was led by a woman at that time of the month? And I thought it was so outrageous, but it made it clear to me that this was not the direction in science I should go at that time. I also began an interest in archaeology and anthropology. And I did volunteer work on an archaeological dig in the Mojave Desert, looking for a population of individuals who had come much earlier than the previously thought groups that had settled in that area. And it was actually a dig that was sponsored by Richard Leakey, who comes from a really outstanding family of anthropologists who had done a lot of studies on early man in Africa. And but I was put in the control pit, which was about 15 feet deep. And I would spend all day long sitting in the control pit doing very detailed work on looking for percussion chips. It had to be the most boring science one could ever do. And even worse, I was not supposed to find anything. So I was in the control pit. And you realize control experiments, it really taught me control experiments are incredibly important. Not finding something is as important as finding something. I also learned in this same experience that science really should be based on facts and observations. And many people, because this dig was sponsored by a very famous anthropologist, it was, a lot of people were really impressed with him and all this. But what I found was that when you asked questions or you challenged anything, they said, well, he said it, it must be true. And that's when I came to the conclusion that you have to be careful in science, and science really has to be based on evidence, and it isn't a cult of personality. So you really, in some ways, science is something where you question authority and you question knowledge. And so, so all these things really, I remember them distinctly, and they were kind of formative moments. As an undergraduate, I also had a number of inspirations, and these were largely people and role models. And so my first introductory neuropharmacology course was given by Huda Akil, who now I see routinely at meetings, and she knows me. But it was the first time I'd actually seen a woman in a leadership position teaching, and she was terrific. And she was somebody who I could aspire to be like, and I continue to do so. She's a wonderful leader, really well renowned. She introduced me to my first series of papers, and these were papers by Julius Axelrod, who actually, he's a Nobel laureate who did his work at the National Institute of Mental Health, where I am now. And but I read these papers, and they fascinated me because they were showing how light impinging on the retina actually could activate cells in the pineal gland, which is a small nucleus very deep within the brain. And that the light activation actually caused a rhythm in the expression of an enzyme in that nucleus. And this really said sensory information from outside can enter the brain and be transmitted back and alter the expression of genes within the brain. And I got very excited about this. And you'll see I later went on into really studying genes. I also at Stanford did my first rotation in a laboratory. And this was, I worked on an enzyme in slime molds, and I was fascinated by homeostasis. There are feedback loops, as you may have seen this morning, and in a talk showing stress, showing how the feedback of all these different hormones alter the activity of enzymes and various things. And we're a wonderful sort of homeostatic organism, which I really appreciate. And so Tag Mansour did this. And kind of unifying all of these three individuals who I really admired, that they all had different stories. Huda came from Syria. She was educated in Lebanon and moved to the United States. She was also, I think, a pioneer in opiate studies of opiate receptor action. Julius Axelrod didn't get his PhD until he was in his 40s. He was a lab technician. And he got the Nobel Prize shortly after he got his PhD, which is an interesting story. Tag Mansour came from Egypt, and he wanted to work on schistosomiasis, which is a liver fluke that really causes devastating illness in Egypt. And so I admired, and I sort of found that their stories were unique and different. And it suggested that even someone like me could consider being a scientist. So that really gave me a lot of encouragement and inspiration that, I should try, go for it. So I ended up going and doing a PhD thesis. And this PhD thesis, I have to say, was a lot of fun. I started working on kind of an obscure problem, but I was interested, I worked in a neuroendocrine lab that studied hormones. And my project was to look to see if I could identify the structure of a precursor encoding the hormone calcitonin, which is a calcium regulatory hormone in the thyroid. So as I was studying that, I was using cell lines and tumor lines that express calcitonin. And I started noticing that some changed their patterns of expression, and this other major protein appeared. And so I began to be curious as to what the other major protein was. And then through a series of experiments I don't have time to explain, we realized that both of these products were being encoded within a single gene. And so at that time, when I started, genes were thought to be collinear with the protein. But soon after I started working, others described that genes are discontinuously encoded within the DNA in the genome. But in this case, these were two separate products, but encoded within the same gene. And through a process called splicing, and in this case, alternative splicing, you could either get a precursor, in this case, one encoding calcitonin, and that was expressed in the thyroid. And then the product the second product that I didn't know what it encoded turned out to be expressed in the brain. And it encoded a small peptide that we called calcitonin gene-related peptide, or CGRP. And this was one of the really first examples showing that one gene could encode two mRNAs and actually make distinct hormones completely by this alternative splicing. So it was really, for me, just the light bulb went on that things, preconceived notions that you have are not necessarily what's there. And in fact, challenging the notion of that one gene encodes one product really led to an interesting finding. And I had a wonderful thesis advisor and a number of other folks who really supported me through that. So it turns out that the peptide encoded by this, and I'm going to do a little aside here, but it was found, I took it to a lab at the Salk Institute, we put it into animals, and it turned out to be a potent vasodilator. And so this simply shows that it causes a dramatic decrease in mean arterial blood pressure in rodents. And that causes a consequent increase in heart rate to compensate for this really big drop in blood pressure. But it was also in a number of pathways associated with pain, and also in pathways in the trigeminal ganglia that were associated with migraine pain. And so I thought I would just give you a little example of how basic science discoveries, and this was discovered in 1982, we showed that it was localized in the brain and trigeminal ganglia and to be a potent vasodilator. I personally left the picture after these green boxes with the basic part, and it was taken on by others. So science is really a community, and it's a community of people who operate across a wide range of approaches, and really it enables one to take a discovery and really move it through and ultimately to become a drug. And so over time, a number of other breakthroughs were made, knowing that this, a little bit about the peptide initially, and having these folks had the good sense to kind of pursue it. Ultimately, it was taken on by drug companies, and now that particular peptide transmitter that we had no clue of what it did turns out to be a major target for migraine. And you see advertisements for various drugs that target this pathway now on TV routinely. So it takes more than a village to move a basic science discovery into the clinic. And what's even more a little bit distressing is that it took more than 35 years for this to happen. So it isn't fast. Technology is always coming along the pike, and so things do move faster now. But this is sort of a lesson in what it takes from basic science to actually getting it to the clinic. My lab went on, and this is when I started my lab at Yale, to study neurotransmitter transporters in the nervous system. And these are really important targets for antidepressants, the reuptake inhibitors, the SSRIs, target transporters that are present on the presynaptic terminal that are responsible for retaking up neurotransmitter after its release. And if you block the reuptake, you end up with an accumulation of neurotransmitter within the synapse. This is also used for treatments for ADHD because the dopamine transporter, methylphenidate, Ritalin, and amphetamine, Adderall target the dopamine transporter. And then drugs of abuse also target the dopamine transporter, cocaine and amphetamines, and the serotonin transporter, MDMA and ecstasy. So I also had a wonderful department chair who was a great mentor, and he really helped me switch into this field, which I had not previously been part of. So it was a great experience. And nobody knew much about these carriers. They thought it might be two proteins. This is an old paper which shows that the binding site for cocaine might be different from the transporter itself. And so we ended up using molecular genetic techniques, some of which I had learned as a grad student, to actually clone the human norepinephrine transporter. And so what was really nice about this is we could identify the sequence of it and begin to speculate about the structure. But the speculations we could make about the structure were that it had 12 transmembrane domains and not much else. But with time, all the approaches for really understanding protein structure and really thinking about these molecules as little machines has just undergone a revolution over time. And so again, building on the work of others really enables fields to move forward. So I'll also bring back in Julius Axelrod, who was one of my initial inspirations. And it turned out his Nobel Prize was actually for initially identifying the activity that was and showing that antidepressants bound to one of these to a transporter molecule within the brain. And so he sent me, just out of the blue, a note describing his previous work and how this built on that. So it was really a wonderful experience for me. So along the way, life intervenes. I married the guy at the next lab bench in my lab. We had a daughter, and we moved to Portland, Oregon, had our daughter, and she grew up. And I want to put this in because life is important too. And you know, I really feel that as much as I've loved my scientific career, I've also found that being able to balance both. And my daughter, I'll say, probably suffered from a mother who was always in the lab. And in fact, wasn't that interested in science at the time, but one Mother's Day, years later, she changed her mind, decided she really liked science, and sent me this card on Mother's Day. And she's now going for her graduate degree in environmental engineering. So she's interested in environmental science. But all of this is to say it's really rewarding. And I will say my daughter is also our best experiment. So we went on to continue characterizing these two families. And in fact, there'll be a familiar face to Ozzy or to Dr. Block here. This is Ethan Block, who spent time in my lab and is now on the faculty at Chatham University. But we continued to work on these. And as you can see, we started, the pictures got a little clearer as to what the structures might look like. We did lots of experiments to identify domains that were important for particular aspects of function. This is my lab in Portland. This is my lab in University of Pittsburgh. And I have to say, I've been so privileged to work with a fantastic group of trainees and colleagues over the years. And I think that you think of science as maybe being an isolated thing. It is absolutely not. It's really a social activity. We work together, and it actually, I think, makes it rich and fun. We went on, and you can see that the structures get a little more finely resolved, and we learned more about these molecules. And I've just really enjoyed. I'm not going to go into details about it, but it's just to say that this stuff gets me really excited, and I really enjoy it. And here we collaborated with a computational group, and we were able to model the actual structure of a trimer of a glutamate transporter, which is another family that we've studied of neurotransmitter transport systems. And we can actually model it, and we can watch for conformational changes. This is a particular conformation that allows chloride to move across the membrane. And so, you keep building on knowledge and learning new technologies and applying them to a problem. And then more recently, we had kind of another surprise for us, which was we had always thought that amphetamine acted by blocking the dopamine transporter and causing release from synaptic vesicles and accumulating dopamine outside. In studying the actions of amphetamine on the trafficking or the membrane localization within the cell of the dopamine transporter and of a glutamate transporter, we actually discovered that there's an intracellular receptor that's activated by amphetamine, and that causes changes in trafficking, and it also leads to signals that move to the nucleus ultimately. The nucleus isn't shown here, and change gene expression as well. So amphetamine has the capacity to do a lot more than we thought previously. So I'm going to have a break from some of my work here and just go through a few things I wish I had known. And I've learned over time, I think. Listen to your data. And I think, I try to tell my students this all the time, really thinking deeply about your results is, one, it's fun, but also it really lets you come up and see new things. So looking for things that others may have missed. And so a lot of for example, the amphetamine receptor was something that we had collaborated on 15 years earlier with another group and totally didn't realize that this is what it was likely to be doing. But later, we kind of, after thinking more about it, we realized that this was in fact likely to be the target for amphetamines within cells. Embracing new approaches. I like to encourage my students to be fearless. Technology drives fields forwards in ways you can't even imagine. And in my time in science, I've seen so many different things and new things that have come along the pike that have revolutionized what we can do. And I think many of you who are now familiar with the new vaccine approaches and the things, those are things we could never have imagined happening a few years ago. And yet, they've been transformational in the way we've been able to deal with the COVID-19 pandemic. So some of the most interesting discoveries come from things that you just didn't expect. And I think I look back and I enjoy thinking about the things that have just surprised me. Don't fall in love with your ideas. Having a hypothesis, having a pet hypothesis that you think is so perfect is probably not a good idea. You really need to challenge the hypothesis. You need to test it. And also not going along with your advisor always. Sometimes I've pushed back on things and sometimes I've been wrong, but occasionally I've been right, and that's usually been a good thing. Read, but not too much. Don't let the literature convince you not to take a risk or do something. Don't believe everything you read, because fields transform over time. Things that you thought were, carved in stone really fall by the wayside as new technologies allow you to think differently about your problems. I always encourage people to ask a question at every other seminar you attend. The every other is because you need to give other people time to ask questions too. Talk to scientists about what you're doing. Communication is one of the critical aspects of science. If you do the perfect experiment, if you make an important discovery, if you don't talk about it, if you don't vet it with others, it will never see the light of day. And so communication in science is much more important than I ever imagined it would be. Talking to non-scientists too. You need to be able to talk about your work to everyone. So, and then writing. We're going to hear more about writing in a few minutes, and write something every day, because communicating your ideas verbally turns out to be an essential element of communicating in science and really driving your science forward. So you might ask, I then switched in, I went to become a department chair and then to work at the NIMH. And I went into a much more administrative. So I wanted to give a little rationale on why I did this. And this simply shows the declining diversity that occurs as one progresses down the academic path. It's for underrepresented groups, it's really embarrassing, and it's something we really do need to work on and change. And I'm very, very committed to that. Even for women in leadership positions really drop off dramatically over time. And it's nice to see women in leadership positions here at your institution. So I think it's great. But I realized that if I didn't do something, and if I didn't step up, it wasn't going to change any faster. So I did, but really inspired by this. I initially did this when I was one of 23 faculty members at the Volum Institute, and I was the only woman faculty member at all. So I think this was a real commitment that I wanted to make. Diversity is incredibly important, and we think of diversity in some ways, and these just list a few of the aspects of diversity we consider, but I also like to consider diversity more broadly. How we think, our language, our background, all of these things gives us a unique perspective on our ability to analyze problems and solve them. And so there are many, many compelling reasons why having a diverse group is important. So diverse organizations perform better on many measures. Diversity is a property of teams. It motivates people to justify their perspective. It avoids groupthink. So you actually are more creative, I think, with a diverse group. And so I just want to say this is what scientists look like. This is my lab before, about a year before COVID. And I think it was one of the most fun groups we've ever had. And really nobody in this group really thought the same about a problem. And it made it a much more dimensional and much more interesting process to do science with a diverse group. Scientists have to do diverse things. I want to just speak a minute to this because I want you all to know that just my model for what a scientist does in terms of looking at how I've approached my career is not all that we need in science. We need people who can do academic research. We need people who can do advocacy. There are jobs in pharma. There are jobs in science policy that are crucial for moving science forward. There's data science, there's technology transfer, and there's really business development. My sister has the same degree as I do, and she ended up going into pharma and biotech and worked in business development. So there's regulatory oversights. We do clinical trials at NIMH. We have social workers, we have nurses, we have many people engaged in this community of science. And I just want to make sure you all know that there's room for a wide range of individuals. And the breadth of possibilities really demands a diverse and diversely trained workforce. So I want to end on just a few minutes, because I don't think I'm going to be able to go through all my slides, but a few minutes about the National Institute of Mental Health. These are our vision and our mission. This is why it's important. And this slide really shows the burden of mental illness relative to many other diseases and disorders. And if you just look at that, at this particular curve, which shows the impact of mental and substance use disorders on the US disability adjusted life years, the impact of mental illness starts early and it just continues and going up. And the impact is huge in terms of the impact on the ability of people to function and lead normal lives in our society. And it's pervasive. So I think it's an important problem, and that there's lots to do with respect. The intramural program is 40 research groups on the NIH campus, 20 basic, 20 clinical. And I think we're unique in the ability to have one group, or like it's sort of a large department, but it actually includes basic scientists like me, and it includes clinicians who are doing clinical trials. And so I've found this to be really fun to really take and think about a basic science discovery and try to move it faster into the clinic. We have a lot of unique expectations, and many of you, this is similar to the program that the NIAID has on the campus, in that we do high-risk research. We try to de-risk research to then have it go out. And I think that's what you've seen the National Institute of Allergy and Infectious Diseases do for vaccine development. Training the future mental health scientists and clinicians is a really important part. We do some creation of national resources. We have a large brain bank for neuropsychiatric diseases. And we try to complement universities and other outside research that's going on. So we try to keep it high risk, high impact, and not overlapping. We have a clinical center, which is the largest research hospital, so we can do clinical trials. We explore the brain and behavioral disorders at many levels. And I was going to just mention a little bit about what some of our scientists do. And I was going to mention depression. We have Carlos Zarate, who has recently focused on using ketamine, which was formerly used as an animal anesthetic to treat treatment-resistant depression. And it turns out it's now been approved, as ketamine has been approved for the use in treatment-resistant depression in 2020. But this simply shows that ketamine impacts the dorsolateral prefrontal cortex, which is a region of the brain that's important in depression, preferentially over the placebo. And so it begins to give us evidence that it's affecting regions of the brain that's important. We also have a group that works on developing neuromodulatory devices, such as transcranial magnetic stimulation and vagal nerve stimulation to treat depression. And where pharmacology acts on molecules and cells, this allows circuits to be targeted with these devices and manipulate the specific circuits. And again, this is a novel and new approach for treatment-resistant depression. And it sort of builds on electroconvulsive therapy, which is effective in treatment-resistant depression, but it has so many side effects that this allows for a more focused treatment, which is non-invasive. We also look at the trajectory of disease, and this is now using cell phones and light trackers and ActiWatches. And this is a study done by Kathleen Merrick-Kangas, where she looked at bipolar patients and studied all these different aspects, having them report daily and at various times during the day on their mood, on their sleep, activity. And there's some interesting outcomes. One, she found that there was a unidirectional association between motor activity and mood. And it turns out motor activity drives mood more than mood driving motor activity, which is quite interesting because it suggests the possibility of interventions that focus on motor activity rather than the end stage, which is mood. I think I'll just go on and I'll end in that I wanted to just give you my final advice, which is choose important questions, find the best approaches to address them, work with people you admire and respect, and work with people who believe in you and believe in your abilities. And I think that's been an important part of my own career. Recognize that there are many ways to get there. So paths are not the same for everyone. Some people go in one direction, then change, and so there are many, many paths. And so if you don't close off an avenue by going in another direction, there are opportunities for incorporating it all. Don't be afraid to fail. I can't tell you how many experiments I've had that failed and how many things that I've had that have failed, but don't be afraid. You learn from your mistakes, and oftentimes failures turn out to have positive silver linings that you hadn't anticipated. So, and also failing means you're taking risks, and in science, taking risks is important. And finally, have fun. I think science can be fun. It's the people you meet, the interactions you have, the questions you can ask, the things you can do, the new technologies that come out really, really are exciting, and it makes it such a fun career. And working with people you admire and respect is really important. And also they should respect you. And so I care a lot also about how people are treated in the workplace. So finally, this is just a list of many of the people who have come through my lab, and I'm happy to answer questions if I have time.
Thank you, Dr. Amara, so much. I think we had one comment that somebody really enjoyed seeing pictures from your lab of the folks that you work with, and I think it really does help to contextualize a story like yours, and I think underscoring, as a social worker, hearing you talk about and underscore the importance of diversity, difference, and what that brings to the work process and how important that is to making a better work process, I think is something that more people are discovering and better understanding. We have one question from a student about, and I think we can probably take one question and then we'll move on to our next speaker, but they wanted to know more about the migraine treatment that you talked about. That was your initial, I think, dissertation research later on that led to that.
Oh, was there a specific question about it?
Well, they just said they'd like to know more about the, what is the drug that's now been approved, or?
There are quite a number of them, and I actually am not even sure I keep track of them now, but there are different ones. So there are they ended up developing small molecules that block the CGRP receptor. There are also antibodies that block the CGRP receptor. And there are also now antibody drugs that bind to CGRP itself and prevent the vasodilation that ultimately leads to the pain. So it's not clear that CGRP is causal, but these treatments seem to work very well. And so there are different kinds. It's the sort of thing you would need to talk to a neurologist about what would be right for you. Mm-hmm. And I'm not promoting them, but they are the newest. And I don't have a patent involved. I work for the government. I'm not involved in it. But I'm happy to see something, to have watched something go from something that was totally, it was, there was nothing known about it, to having something that really is useful in the clinic, and to see it emerge as being a really useful treatment. And also it was fun to watch the experiments that followed, which are, how did they figure out that it was really going to be useful as a drug? And those things are kind of interesting. But there are kind of three different flavors of this drug. And actually when I started, there were no options for antibody drugs. So that wasn't even a possibility. And so neuropeptides, they were often difficult to target because you had to find just the right small molecule that would fit in the same binding pocket, but it didn't really look like the peptide. So so now with antibody technologies that can be used, it's made that more amenable for as a treatment.
Thank you, Dr. Mara. We'll have a few more questions, I think, that'll come in, and what we'll do is at the end, but as time allots, we'll take those questions and have you address them. So thank you so much for your presentation. I'm going to take a moment now to introduce Sarah Elaine Smith. So she's our second keynote, and I think the great thing about our conference is that we're really doing a wonderful job of blending together sort of different forms of research, creative expression, and scholarship. So Sarah Elaine Smith is an accomplished author who grew up in Greene County, right nearby in Pennsylvania. She invites you to learn more about her books and inspirations on her website, sarahelainesmith.com. So Sarah, I'd love to give the floor over to you, and I will mute and disappear.
Thank you so much, Dr. Block, and hello, everyone. And also thank you so much, Dr. Amara. That was a fascinating presentation, and I don't often get to travel in, like most of the time whenever I give presentations, I'm in a room of other novelists and poets, and there was so much that sparked for me in hearing your presentation. In fact, the statement, finding nothing is just as important as finding something, is like really tasty to me and will be sticking with me for a while. Before I get started, I just wanted to thank everyone for being here. We're all on our computers so much that it really, it takes something to get on the computer again, I think, in these days. And I'm really grateful to you all for being here. And I also just wanted to shout out really quickly all of the poets who did presentations of their work as part of this conference. Bryce, Chris, Sierra, and Derisk. I checked out your work and I just loved it. And I found it so exciting and so hopeful that during the pandemic, you've produced such spectacular work. It really gives me so much of a sense of optimism whenever I see that we're all able to sort of digest and be in the world using art as a sort of lens and a transformative experience. And I was just tickled by your work. So if you're watching, just a little shout out to you. All right, so I'm going to present to you all now. Let me see here. All right, away we go. So the question that I was posed in being asked to speak here was to talk about the nature of research in my work. And the way that that question comes up organically most of the time in what I do is in the question at Q&As, where do you get your ideas? And so just by way of introduction, hello, this is me. I'm the author of Mary Lou is Everywhere. This is the book, hooray. And also a poetry collection called I Live in a Hut. And I have studied poetry and fiction at the Michener Center for Writers at the University of Texas at Austin, and also at the Iowa Writers Workshop. And currently I teach creative writing, although I have sort of taken a path away from academia and toward teaching in sort of alternative online spaces. And along with that detour away from academia, I found that my work and my teaching focuses on the intersection of tarot divination, somatic experience, and alchemy, which I'll talk a little bit about sort of, here and there in the margins. And the way that I approached this question, at first I thought, do I really do research? Because my sort of default definition for research is this, what I have on the slide here, a systemic investigation and study of materials with the goal of arriving at statements of fact or developments of theory. And it's that notion of arriving at a statement of fact that I was sort of like, I don't know if I do that. And of course, there are lots of writers who do research in this mode. Specifically, historical novelists, you might consider. There's a really wonderful book called Isadora about Isadora Duncan's life, which is incredibly research driven. And also in poetry collections, for example, Layli Long Soldier's Whereas, which in addition to using lived experience, uses a really an interesting sense of legal language and sort of governmental promises and discords in communication with First Nations people. So research of this sort of fact arrival nature is not uncommon in creative writing in any regard, but it wasn't something that I really identified with. And so for me, research can also mean a study of life with the goal of arriving at questions and possibilities. So instead of facts, instead of seeing it as a sort of encapsulated process that arrives at something stable, it can become this sort of open-ended parentheses upon parentheses upon parentheses, arriving at new questions and interesting possibilities. And this picture I've included, this is my cat Falcor and I doing a tarot spread. Tarot is an image-based divination practice, which I've found has a lot of resonance with the way that I think about creating poetry and creating fiction. And, you know, for a long time, I started off as an undergrad at Carnegie Mellon, where I was a double major in the critical theory track and the creative writing track. And for a long time, I was really interested in that idea of study as knowledge production, which was one of the means of engagement that was really modeled around me. In other words, writing research papers and publishing them. And as I've sort of gone along, I've started to incorporate more inquiry into the nature of what occurs day to day in my life, inquiry into what comes toward me from lots of different directions, and considering that a kind of research. And so what I'm going to do today, backtrack. I wanted to give you a little bit of context for thinking about creative works as possible answers to what if questions. So for example, and I've just given you, these are a few examples from some of my favorite writers. Octavia Butler's sci-fi novels, The Parable of the Sower and that whole series answers the hypothetical question, what if climate change created social instability that drove us from our homes, which might not be so hypothetical soon. Who knows? That book has been hauntingly prescient lately. Or Roberto Bolaño in 2666, what if a novel investigated an enigmatic writer by examining the relationship that critics have to that writer's works? Or Terence Hayes, who is a fabulous poet, what if blues tropes were applied to persona poems? And if you're curious about that, I suggest you check out his book, A Wind in a Box, and particularly the persona poem, The Blue Cool, which is a blues poem from the voice of Kool Keith. It's fantastic. So in this spirit, research becomes, for me, a process of finding more questions. And I have to say, where do you get your ideas is probably the number one question that I hear in Q&As after readings of my work for poetry and for fiction. And for a number of years, I was actually pretty salty about this question because I couldn't relate to it. It seemed so obvious to me that the world is full of fascinating conditional ideas that can take form as poems or as narratives. And I used to give this really sassy answer. I would say, there's just a Time Life subscription. It's called the Book of Ideas, and you just get it in the mail and you pick them and whatever. Because, in some sense, it just puzzled me so much that this process wouldn't be innate to everyone. And so I'm going to kind of give you an example of a germinal idea and the way that it traces through my thought process and through personal experience to become something else. And in this case, particularly, I'm going to give you an example of something that helped me end my poetry writer's block, which I don't know what happened. I had a really horrible case of writer's block, specifically about poetry, the more that I became a novelist. And, I didn't know really how to get into that. If you've ever been blocked as a writer, you know that it's a horrible experience. And for me, the way that that shows up is I sit down and I try to, I look at the blank page and somehow there's just nothing to say. Or even worse, I write a line, and then I evaluate it and think that is just too horrible and stupid. I have to delete it right away. So for a long time, for years really, I had this feeling of distance and closed offness and sort of finality around writing poetry. It just seemed like a channel had been closed, a door had been closed, and I was on the other side of it with no idea how to get through. But the new source of ideas that I eventually found that helped me find my way back into writing poetry came from a really surprising source. It started with the style sheet from my novel, Mary Lou is Everywhere. And this is sort of a fun behind the scenes look as well for you if you're interested in publishing and you're hoping to publish a novel yourself someday. This is something I didn't know myself, that before publication, a novel is fact-checked, even though it's fiction. Fact-checked in the sense that anything about the world as it's represented in the book, if it corresponds to our world in some way, it ought to be, names of places ought to be spelled correctly, anything like that. And I found that my book, which takes place in Greene County, was often kind of confounding to my publishers because it included so much regional specific language, things that we say in southwestern Pennsylvania that people don't say in other places, or tiny towns that are so small that people haven't heard of them. In fact, I remember very clearly one of the fact checkers for my book getting in touch with me to say, there is no town called 100 West Virginia. And I thought, I know a lot of people in 100 West Virginia who'd be very upset to hear that. But in a sense, part of the publication process is taking this fictional world and bringing it into this reality. Even though it's brought into this reality in the form of a book, it has to make some kind of sort of like transactional agreement with our world, with the facts of our world. And another way that that takes place is the style sheet. And what I have here on this slide is just the very beginning of my style sheet, which is sort of like a list of, these are all of the words in my book that are used in unusual, incorrect, non-grammatical formations. And the reason that they're included in this word list is as a note to copy editors not to change them. If you happen to see Kogos with 2 initial caps, in other words, that's correct, don't change it. It's an in-house document for copy editors to make sure that they respect the way that the author intends to use language, in other words. And so some other examples of things that end up on the style sheet are maybe some, ways of speaking that you might recognize if you're also from Northern Appalachia or Greene County, or really any part of our region. We say slippy instead of slippery. It's snowing outside, so it is pretty slippy today. We say crick instead of creek, and needs done instead of needs to be done, all of which are noted on my style sheet. And if you're interested in seeing the whole thing, by the way, I have a whole essay about this on my website that I encourage you to check out. And, as I've shown on this slide, sheets, of course, is on my style sheet, really important part of our regional culture. All of these things are kind of like a fingerprint for our place and our ways of speaking. And actually, I'm going to read you just a really quick little excerpt from the book. And this is a passage that I think, if out of everything in the book, this is the passage that people come up to me and talk about the most often as something that really stuck with them, that really meant something. And in this passage, the narrator, Cindy, is describing what it's like to be from Greene County, some of that identity of place. We were people who were bent into catastrophe postures by poverty, black lung, heroin, WIC vouchers, fluoride, Miller time, a caustic species of aloneness, perfectly well-intentioned social workers, post-industrial blight, single-A football, pepperoni rolls, and things like that. In a sense, like that passage is giving you just sort of an encapsulated version of this effect I'm talking about here, in which the style sheet notes all of those things as special cases, which are to be preserved in this text instead of standardized. And, you know, the thing is, this is a really kind of mundane part of the publication process. It's something, you know, fairly administrative that you do in the process of having edits made before publication. But I found that it made me really emotional. And that is where this process of research begins for me, in a sense. I ask myself, why does this provoke an emotional reaction in me? Because it seemed surprising. And it wasn't something that friends of mine particularly noted. And so I realized that the thing that felt so emotional to me was that this way that I grew up talking was being deemed correct, worthy of protection, worthy of inclusion, considered part of the reality of this text in this world, which was notable to me because when I left Appalachia to go to grad school, that was the first time that I noticed really that people made fun of things that we said. And for example, I didn't know that needs done was an incorrect grammatical construction until years later when I was a proofreader at a newspaper. And this was a really sort of disheartening experience because I had no idea in some ways. It's almost like being able to overhear a conversation that's about you, but no one knows that you know that it's about you, because I was, you know, in conversation with other educated grad school people who presumed, of course, no one here is going to use words in this strange way or speak ungrammatically. This is about someone else. And I felt this sense of belonging and this sense of shame, being excluded, even though I passed. And I still don't, you know, you might notice I don't have a really intense Appalachian accent, and it's because my parents are both from the Midwest. But it was a really strange experience and really disorienting because I didn't even know sometimes how to speak correctly. You know, if you don't know that something is wrong, then you don't know. You're sort of inside this other reality. And it felt so validating to realize that in this book, in the act of writing about place and being so specific, I had found myself in a situation where that way of speaking would be protected. And so, oh yes, to go back. Oh no, I can't go back. Nuts. Sorry, everyone. I'm going to escape out of this and go back because this is important. Right, here we go. Part of the realization was asking myself the question, what does that remind me of? Which is another really important one. Usually whenever something is important, it adheres to some kind of pattern. There's some echo, there's some thought loop, it coheres with something, and that usually leads me to other interesting questions as well. And so this sense of some kinds of speech being collected and protected, I asked myself, what does that remind me of? And I realized it reminded me of our style guide at the newspaper, the Austin Chronicle, where I worked for a few years as a proofreader. And if you've never worked on a newspaper or at a copy desk, I'll tell you, a style guide is sort of like an auxiliary dictionary for your publication. It is, includes all of the terms that, like the style sheet, might not be correct in the dictionary, or they might not be sort of conventionally correct uses of language. They might seem at first like errors, but they're not errors and they're worth being protected. And I have some examples from the Austin Chronicle style guide. A nudie suit, which, in Austin, it's a big music town, big music scene. And the paper covered music often, obviously. And a nudie suit is a sort of Western wear staple, this like cowboy suit that has all of this sort of phantasmagorical embroidery all over it. And it's called a nudie suit because the creator was named Nudie Cohen. But if you just saw nudie spelled that way and you didn't know otherwise, you might say, well, that's wrong, but it's not. And similarly, Rocky Erickson, who is an Austin native or was an Austin native and the lead singer of the 13th Floor Elevators, was a really important and central part of Austin's music scene. And as you can see, his name is spelled Rocky without a C. If you didn't know better, you would think that's incorrect and I'm going to fix it. But it was important for the newspaper to have this auxiliary dictionary because our authority and our belonging as a regional newspaper depended upon our correct usage of these terms. It would be considered, a worse mistake than including a comma splice someplace in an article to misspell Rocky Erickson, because it would bring into question, do these people really know where they are? Are they really a part of this culture? And that's why it was so important for us as proofreaders to have access to this information. And this was a real discovery for me. It made me realize that in so many ways, our sense of a specific culture comes from knowing edge cases and unusual ways that people speak. And that possibly I was having this emotional reaction because it was granting authority and a sense of reality to this place and its ways of speaking and its general lived reality, giving that a sense of belonging and authority to a way of speaking that's otherwise marginalized. And that, I think, is why that passage that I read is something people come up to me and talk about so often, because they recognize themselves. And who knows what it is about being seen, but you can see that this is an incredibly important part of our culture, even just in memes. Our memes are so often about being seen, even if we're being roasted, in a Virgo starter pack meme or whatever. Whenever we see something like, oh, pepperoni rolls, she gets it. We also get to feel included in the literary canon. And so what had happened here, and this is where this idea bridges, it begins to sort of leap from this context, the sort of grammatical context that it originates in, into another creative form. I realized that my world had been given a lexicon, and a lexicon is a vocabulary that is specific to a place or a body of knowledge. And in this case, specific to a place. You might hear that term also, in other fields, in scientific fields, in linguistic fields. But in this case, it just meant this is a specialized vocabulary and that it is at least as important as the dictionary sense of what is correct. And I loved that idea, thinking about a dictionary, a lexicon of this place that I'm from. And so I began asking myself, how else can I use this idea? And that is another question that is an incredibly important part of my idea research process. And I've given you a few examples here. What would the lexicon of an imagined world consist of? What if I reverse engineered the process by making a lexicon first and then making the text second? Basically just reversing the process that I had noticed in the, sort of publication and edit process for my novel. And that is exactly what I did. I decided to make a lexicon for an imagined world by gathering interesting phrases from texts that are not poetic or not meant to be poetic. Texts like stargazers guides or nature guides. Nature guides are full of incredible language because it makes sense, our sort of first interactions with flowers and birds and all of the beauty of the natural world, we tend to give all of those things really colorful, wonderful names. And so the question that I was posing here is, what would happen if I wrote poems from this starting point, this made up lexicon? And this lexicon is something that, I sort of add to it from time to time. It's just like a living document of phrases, words that have a little bit of a spark to them, things that have some of that evocative power, in the same way that, in describing our region, single A football and pepperoni rolls snap. You see something, you get an image from the collision of those phrases. In a similar way, I collected all of these phrases that have that capacity to spark against each other. And what I discovered is that in doing so, and making this lexicon and sort of doing everything in this reverse order, I overrode that feeling of sort of numbness and shut downness that I had been feeling around writing poetry, because the first idea didn't have to come from me, in a sense. The first idea could come from imagining the text that would be reflected in this lexicon. And I've written so many poems this way. It's so enjoyable and so much fun. It really has sort of taken me out of that feeling of incredible, self-serious stress about why can't I do this? It makes it so easy to approach. And so I've given you an example here that has a few of the phrases or words that you had seen in the previous slide. And in a way, these poems that I've been collecting, and I've written almost a book's length collection of them by now, while they're all invented and they're all in various forms and use the sense of various speakers, they cohere because they have this capacity to refer back to a feeling of a real world boundaried by all of the things included in this lexicon. And the funny thing about all of that is that in so many ways, what I've just described is such basic writing advice. You've probably heard show, don't tell, and use concrete, specific details. I myself have heard that many times. Those are two things that I teach in every single class I teach. And it's so humbling in a way to realize that this inquiry process takes me through so many interesting ways of thinking. And often it delivers me back at my doorstep, right where I was before. Which gives me the sense that what I'm really doing here, instead of traveling on a sort of, one direction upward, upward and onward progression, I'm traveling in a spiral, in a sense. I'm moving around these ideas, seeing them from new angles, and then returning to familiar places and familiar ways of thinking. And that is something that I've found to be, as I said, really humbling to realize. These ideas go back to some of the very first things that I've learned as a writer. And in some ways, what happens for me as part of the research process is not so much that I discover new things, but that my questions take me into different vantage points for looking at what I've seen before. And that, I think, is part of the function of question asking as a primary process for me, because what would happen, and I think part of the reason that I had such terrible writer's block for so long, is that I had forgotten about questions. I had forgotten about possibility. And instead I had it's no coincidence that this happened after I published my first book of poems, I think, because at that point I had decided, well, now I'm a poet. Now I'm a published poet. Now I have to write a second book. What's it going to be? Those questions were sort of closing me down in some way. Or maybe it would be more accurate to say that I was looking for answers and closing off the possibilities, which for me as a writer, possibility is it's the whole reason I do it. It's what makes me excited. And whenever I forget that, I sort of lose my way a little bit. And so to finish up, I want to talk about how you might be able to apply this idea of working to your own work, regardless of what your work is. And this is one of the reasons, one of the things I love so much in Dr. Amara's presentation, a similar focus on the importance of the question, because the question is sort of like the scope, the sort of field in which you're going to look is determined by the question, and a narrow question can produce narrow results. And so the thing that you might consider if you are looking for some way to sort of energize your practice or shake up something about your own research, your own work, is simply by asking questions. And I've collected here, you probably noticed them as we went through the presentation, because I marked them in bold, some of these questions that I returned to. What does this remind me of? Who would I be if I already knew the answer? Why does this provoke an emotional response? Where have I seen this before? Oh, and one of the most important ones, is there any way the opposite is true? Because often what I have in place of a fact is actually just a belief. And whenever I ask myself, is there any way, even a very small way, that the opposite of what I think I'm seeing is true? Typically it is. And that possibility is really fascinating. And I also want to add here, because I think this is an important note about the authority of a lexicon, the authority of a dictionary, the authority of a sort of central understanding of what is considered correct. Asking questions about that is also an important social justice practice. And you might apply some of these same questions to looking at society, at the place that you live, at what's going on in your neighborhood, who gets on at the bus at what stop. What articles are they talking about on the cover of the Pittsburgh Post-Gazette versus the Pittsburgh Courier, which is a Black-owned, Black-written newspaper? These questions are not just a way of engaging with creative work, but a way of engaging with the world. And when I begin with a question instead of beginning with a statement, I get to travel in that world and see a lot more. And the two that are the most important, at least in my, my sort of take, is what does this remind me of? And why does this provoke an emotional response? Because, it can be all too easy to avoid emotions in favor of thought when really those things can be brought together. Especially because the mind, at least in my experience, will always answer a question, even if it's absurd. Like, what would it feel like to have wings? The mind will produce an answer, whether you've had the experience or not. And that is the beauty of the writing process. So I want to thank you so much for being here. And I do encourage you, please stay in touch. If you have anything that you'd like to talk about as a sort of spin-off of this, please email me. I'll be so happy to answer. And thank you so much for your time.
Thank you so much, Sarah. That was really wonderful. It made me think a lot about this word in my family's lexicon, which is the word boons. We use this word. It derives from my dog and the way she would behave sometimes. She's no longer with us, but my child asked me the other day when I said something was like, there was a boons. He was like, is that a real word? Because I've never heard anybody else use it. And it just made me think a little bit about that creative expression and how, maybe there are like you're talking about with Appalachia, that there's a language there, but it's also like unique in our own families as well. And what a spirited and wonderful thing that is to have that uniqueness of words to share together.
Yeah, absolutely. It's a way that we show love for each other, I think, to learn the
Definitely.
lexicon. Yeah.
Definitely.
And to develop one together is like the way that we have relationships with each other.
Yeah, for sure. I want to invite everybody, if you'd like to add questions into the Q&A for Sarah, I'm going to move us along to the presentation of the awards. So I'd like to give the floor over now to Dean Kristen Majoka, who's the Dean of the College of Education and Liberal Arts here at California University of Pennsylvania. I believe Dr. Ross is also with her, who is involved also in the Linda and Harry Serene Leadership Institute. So the floor is yours.
All right, hopefully you can see my PowerPoint presentation just fine.
Yes, we can.
Very good. Okay. Thank you so much, Ozzy. Thank you, Dr. Block. Thank you to our speakers and presenters. I very much enjoyed them and had many of my own thoughts and my own experiences as you were talking and presenting your research and your work. Very thought-provoking. Thank you. My name is Dr. Kristen Majaka. I'm the Dean of the College of Education Liberal Arts, and I have the pleasure of announcing our Strike a Spark Essay Contest awards, winners, and honorable mentions. So first, I'd like to start by telling you a little bit more about the Institute. Linda and Dr. Harry Serene are both California University graduates, and they founded the Linda and Harry Serene Leadership Institute. And the purpose of the Institute is to give back to the university, but more importantly, it's to give back to the local community. The purpose and mission is to create and maintain a comprehensive leadership development program that integrates academic and experiential learning through study, workshops, and also empowerment. And it manifests itself through participatory leadership. Their focus includes women, minorities, and international students as well. Since 2019, Linda and Dr. Harry Serene have sponsored many speaker series, scholarships, workshops, leadership conferences, and in the future, they do anticipate offering organizational management workshops and professional training seminars. So we thank them so much for supporting our students and the future leaders that CalU produces. This year's theme for the essay contest is staying connected through leadership in research and creativity. The Serenes believe that this past year has presented great challenges to all of us, but in particular from a leadership perspective, and that couldn't be more true. The students who presented and sent in their essays for judging, I thought it's tough. It's tough to produce 4 to 5 pages in an academic format on top of all of the other studies that you're doing. But I couldn't have been more proud to have read the essays this year. There were a variety of approaches on the topic. Some of the approaches were primarily research-based, some were primarily narrative-based, but some of them also included a research and a narrative combination, which was really nice. I enjoyed reading all of the essays, but today we are highlighting some of our essays, which will be our honorable mentions, and then I will get to our top 3 winners. So I'd like to announce and congratulate our 4 honorable mentions this year. Bridget wrote an essay where she discussed how to sharpen leadership skills that result in tactful, loyal teams that create, that imagine, and collaborate even through a computer screen. I loved reading your essay, Bridget. Thank you. Emily, she presented earlier today, and her work is always spot on and grounded in strong research and best practices. Her discussion was just a lovely conversation about creatively building rapport, which I know Emily, and she does that very, very well. Tamara, she talked about her experiences through school and through sports and how that helped build leadership and helps prepare her for leadership roles in the future. Again, a really nice blend of both research and narrative. And then Taylor. Taylor explained the value of touching hearts and minds through resilience, through agility, through listening and compassion. I also know Taylor, a very compassionate young woman. Congratulations to our honorable mentions. This is an award and a recognition that you should put on your curriculum vitae and your resumes. Congratulations. Next, I would like to introduce and congratulate our 3rd place winner. Our 3rd place winner is Otis Jordan III. Otis wrote a lovely essay with many narratives about his grandfather, and he addressed the challenges facing African Americans through a situational approach to leadership, through conclusivity, through honesty, trust, transparency, and integrity. Congratulations to Otis. Our 2nd place winner, Sheila Renz. Sheila's essay was thoroughly researched. It was an essay about virtual hearings, alternative dispute resolutions, and online dispute resolution services. It was a wonderful integration of the studies that Sheila is engaged in here at CalU, and it was a well-written essay. And Sheila is our 2nd place winner. Congratulations, Sheila. And without further ado, our first place winner is Michelle Sable. Michelle Sable wrote an intriguing, well-researched, well-written essay about a search for creativity and meaning in artificial intelligence, online learning environments via a phenomenological aspect. Couldn't have been more timely. And again, an excellent essay. Congratulations to Michelle. Thank you to everyone who submitted an essay. Congratulations to our honorable mentions and to our first, second, and third place winners. I'll be contacting you a little bit later today to get more details from you on how we can get, what's important to a student is that check, the award that you won. And again, we want to thank the Serenes and the Leadership Institute. It has been my pleasure, and I look forward to the rest of the events with Strike a Spark. Thank you.
Thank you so much. So I'm going to take a few things from the chat now. I know we just have a few minutes remaining. Dr. Fasanik made a comment. She's actually running the 1 p.m. Digital storytelling for any of you interested in attending that wonderful course. But her honors 250 students read your book, Sarah, and just wanted to give a little shout out to them. They really enjoyed it and really enjoyed engaging with that. And I did have a question from a little earlier for Dr. Amara. So Dr. Amara, if you're available to respond to this, the question was, what do you think about the MDMA assisted psychotherapy protocol that MAPS is doing?
Yeah, I think there's been a lot of renewed interest in MDMA and psychedelics as well, to use them in combination with psychotherapy and other approaches. I think the data really hasn't come in yet in terms of how effective the treatment is, but I do think there are new approaches to really analyze that, and I think it's quite worthwhile, and I think there's a lot more activity on those areas. My own lab studies MDMA, but much more at a mechanistic level, but the idea is if you can understand how it's working, on particular groups of neurons in the brain, you might be able to find other ways of manipulating the same system. So I have no preconceived notion about it, but I think it's definitely a lot of renewed interest in those areas and worth pursuing.
Thank you so much. Any other questions from our audience or panelists, questions for one another? I think it was a really phenomenal sort of, what do I call it, potpourri of things together. I want to blended. I heard somebody talking yesterday about a bouquet. And I think like in some ways, maybe that's a better description than potpourri, because this was like, it all went well together, but each really was a different thing. And I think really underscored the different ways in which students can hear a story about somebody's path, hear about the ways in which research is being engaged in, both, in creative expression as well as in like a more, basic science research capacity, but the different paths that got us to these places. And I think that's such an important thing for students to really hear. And I don't want to say necessarily be inspired by, because I think a lot of them have gotten to this place already through other inspiration, but to kind of give them that charge to keep pushing forward and to seek that, that place, Dr. Amara, you talked about it, the fork in the road, that there are many different paths one can take, and each one of those is valid. And, just because you choose one fork doesn't mean other forks won't open up. And I'd say I have that from my own lived experience as well in my higher ed education and career path. So I just want to say thank you so much to everyone. Really appreciate the time spent and, sharing with our students. And I will share out the YouTube link to all of you as well, so that you can share this, the sort of constant place that this recording will stay. So thank you, everybody.